Dr. Karl Kieburtz, who is a professor of neurology at the University of Rochester and the founding director of the Center for Health and Technology at the University of Rochester, which contains the Clinical Trials Coordination Center, which we're all familiar with. Karl also founded Clintrex Research Corporation in 2008. This is a company that advises on the scientific and regulatory aspects of drug development for CNS disorders. And so it's just my honor to introduce Karl, who will give us our first talk, 30 Years of HD Clinical Trials, where we've been and where we're going. Karl, thanks.
Thank you. Good morning. I only have 2 slides, and I'm not going to show them for a minute. I just want to, at the risk of almost being solipsistic, I'm going to talk about my personal journey in Huntington's disease and try to use that as a guidepost for talking about what's happened. So almost exactly 43 years ago, in October of 1980, I found myself in a room alone with a person with Huntington's disease, a stopwatch, a pencil, and a stack of paper. How did I end up there? So a little background. I spent the summer before that time, which was between, I just graduated from Amherst College with a degree in neuroscience. Just to give you a little flavor of the time, I was in the first freshman class that admitted women to Amherst College. And at that time, it was the only undergraduate liberal arts college that offered a degree in neuroscience. So that's how I ended up there. And I spent the summer, and I spent time doing like Ungerstedt rat model stuff and in the lab. And I liked neuroscience, but I didn't like doing science. So I spent the summer, my grandfather and father were gold rushers, prospectors in Alaska. Grandfather and great-grandfather, not my father, at the turn of the century. And so I went back and I grew up in the Pacific Northwest. I spent the summer living and hiking in the Pacific Northwest and Alaska and trying to figure out, did I really want to go back east again to a flat farmland place called Rochester? Is that, or did I want to stay in the mountains and prospect for gold, so to speak? Well, I went back to Rochester, but I met with my advisor and said I wasn't really interested in doing something simple. I really wanted to prospect for gold. And he said, oh, that's interesting. I have something for you to do. And that's how I ended up in that room. I became a neuropsychological tester in IRA's baclofen clinical trial that was ongoing at Rochester at that time. So that's kind of interesting in a way, because think about where we are now. I knew nothing about clinical research. I didn't know much about simple digit or Stroop either, but had been taught, and we were capturing everything on paper and pencil and entering it in mainframe computers at single sites. That was kind of the state of the art 43 years ago. So I'd like to talk a little bit about, from my experience, what happened after that. So if I could have the first slide. Ira and I've known Ira for a long time, essentially since then. So you might not be surprised that we kind of formulated things a little bit similarly. Down the left-hand column is the decade, 60s, 70s, 80s, 90s, and there's columns here about technology, FDA, Health and Human Services, United States government stuff, and HD. So one thing I learned about prospecting, there's kind of 4 things you need to be a successful prospector. 1, you need persistence. 2, you need to know your territory. 3, it's almost impossible to be successful on your own. You need joint action. And 4, you need the right tools. So this is kind of the core of being successful at prospecting. Prospecting, by the way, is finding hidden value. It's really the core of prospecting or mining for gold. So those themes can kind of touch through here. 1962, Kefauver-Harris Amendment. By the way, how many people were not yet born in October of 1980 in this room? Come on. Yeah, most of you, right? So this seems like ancient history. I'll just say, so 62 was the Kefauver-Harris Amendment, which was the first time you actually had to show evidence to get a drug approved. 76, the Belmont Report. This is, the foundations of autonomy, beneficence, and justice, the idea of informed consent, and and the balancing of risks and benefits. 1970s, by the way, mainframe computers. When I went to Amherst College, the entire basement of the administration building was occupied by the only computer on campus. That was the state of affairs then. 20 years later, I was working with Ira on the Parkinson's Study Group DataTop study, and one of my first things was to look at adverse events, 4,000 of which have been recorded, 25% of which were coded other. And my job was to figure that out. And I sent him a memo about it, and he said, nice job, but don't send a memo. I need you to start communicating with me, this is 1989, with something you've never heard of before. It's called email. That's not so long ago. The World Wide Web, only 30 years ago. The Genetic Information Non-Discrimination Act, introduced by Louise Slaughter into the United States Congress, Congresswoman, the former Congresswoman from Rochester region, 1995. Accelerated approval, spurred primarily by HIV epidemic, approved in 1990, not approved in 1992, but it became a standard. What's happening in HD? Ira already alluded to some of these things. Marjorie Guthrie's stimulated Commission to Combat HD in 67, a presidential commission with President Carter in 77. HDSA formed in 83. The Baclofen trial that I alluded to started in the late 70s. The Huntington Study Group formed in 1993. Let me just say about so little Huntington Study Group, which, by the way, Sherry, was formed before the gene was identified, not after. First meeting consisted of single-digit number of people in a conference room in the Hyatt Bethesda over lunch. Prior to that, all HD research primarily existed at individual institutions who didn't really like each other. To a certain extent, Vancouver, Baltimore, Boston did things independently, differently, and better than one another. So the idea of getting everybody in one room was a bit unusual. I had also been working very hard on Parkinson's Study Group things, and one day Ira said to me, you know what we should do? And I thought, quit or do less. He said, we're going to form the Huntington Study Group. I thought, he's out of his mind. How can we start another thing? It's like it's too busy now. So vision, persistence. UHDRS 96, you may not remember that the first function you could do to become a member of the HSG was to complete forms. Yes, paper and pencil forms. And the first forms we completed, does anybody remember? The QNE, the Quantified Neurologic Exam. We started off with that. And how did that go? Terribly. The sites hated it. So we adapted and we said, well, maybe we need to develop our own scale. Ira passed that off to me, so I worked with Jack on the motor part, Peter Como on the cognitive part, both of course whom have both passed since then, and Ira on the functional part, putting it together, and then the idea was that you garnered sweat equity by completing a UHDRS form, I believe. Once it was promulgated after 96 that you got invited to the HSG meeting, expenses covered if you had completed 10 UHDRS forms in the last year. So Times were easy then. Intro HD, OPC 14117, an Otsuka antioxidant. Study kicked off in 1996. First multi-center trial in Huntington's disease in the history of the world. Unbelievable, huh? CARE HD started in 97, first NIH-funded multi-center study in Huntington's disease. Happened to be the PI, so I can remember we enrolled 340 patients at 23 sites in 11 months. Speaking of joint action, that cooperative effort, that interest, was because from the very beginning, the HSG involved not just, as Ira alluded to, basic researchers, geneticists, clinical researchers, but also patients and families in the advocacy organization. So we're much better able to work together collaboratively. So that was a lot to get done in 40 years. But look what happened in the next 20 years. If I could have the next slide. A little harder to read. Just tech context, iPhone introduced 07, social media, X, Meta, all that didn't exist before 07. Only slightly more than 15 years. Now they run our lives. GINA passed, speaking of persistence. 6 Congresses later, it passed. Tetrabenazine approved. Very important in the tool generation world, the HiQ Foundation and CHJ. I collaborated with the University of Rochester and the Huntington Study Group to create the first academic CRO-like structure that had electronic data capture. This was like changing the tires on a car going 60 miles an hour. The CTCC had to shift from the old paper and pencil, 2-part non-carbon paper. We used to do 2-part NCRs, is how you did data collection. That all happened. Why is that important? Because it really triggered the ability to have efficiencies in the conduct of trials. And what you see here, NIH and FDA-funded trials in the noughts of riluzole, minocycline, phenylbutyrate, futility study of minocycline, 2CARE and CREST-E, which are long phase 3 studies of nutritional supplements, CoQ and creatine, and Prequel, launched in 10, the first clinical trial in non-manifest gene carriers in Huntington's disease. All that happened in that decade. Prior to that, 2 clinical trials in Huntington's disease, Intro and CARE HD. Simultaneously, industry-sponsored trials started to happen. Here, these are primarily sponsored by Neurosearch, Heart and Mermaid with the drug we now know as pridopidine. Tetrabenazine trials were conducted. Diamond and Horizon were trials of Dimebon by a company called Medivation. Just a little story about Medivation, put things in context. Have you ever heard of Medivation? Yeah, sure, remember that? Medivation, I got a call. I remember I was having lunch at my aunt's house from Lynn Seeley, because I happened at the time to be the chair of the FDA Advisory Committee and chair all the DSMBs for the Alzheimer's Disease Cooperative Study about what did I think about a study in Alzheimer's disease. And she told me what the drug did and what they had done. And I said, I think you should look at Huntington's disease. And so they did, eventually. When the Horizon study failed, Medivation's stock went up 30%. Now, that's interesting. Why would a company's stock who's investigating Huntington's disease go up 30%? Because they were also developing an oncology drug, and the markets decided if they finally stop trying to treat Huntington's disease, the company's going to be worth more. By the way, they eventually sold for $12 billion to Pfizer for a unique anti-prostate cancer drug. So industry was involved, small companies, very tentative, high failure, hard to get other companies interested. But the bug was kind of out. So what happened in 10, Research Kit released by Apple, meaning that we started to use these miraculous smartphones as data collection devices. We started doing video visits. digital sensors. Suddenly, clinical research, which has largely been a luxury item, people have to have the wherewithal to actually be able to come and participate in research, is starting to be democratized through the distribution of data collection tools on kind of a ubiquitous internet-based basis. And trials really took off on an industrial level. I'm not saying everything, so apologies if your studies are not on here. These are just things I, one way or another, directly touched. So Novartis anti-chorea drugs, REACH was a metalloprotease inhibitor, first in ARC around deutetrabenazine, and Austedo was approved. Pride, Legato with pridobidine and laquinimod. Signal, first infusion study, monoclonal infusion study in Huntington's disease, rapidly enrolled, big uptake by investigators and patients and families. Roche Ionis. Now we're starting to get into things that are really starting to target the nub of the matter, the gene itself, or, in the case of Azavan, trying to start to address other aspects of Huntington's disease and motor. Proof and Connect started in that decade as well. And more recently, we have an exon with another, an exon with another monoclonal antibody at a different kind of mechanism. Wave coming through back again after some initial difficulties. Speaking of persistence, Roche, ditto. PTC with another mechanism, SAGE, looking at cognition, and there's an N-acetylcysteine study being conducted in Australia. So there's kind of this distribution of the effort on a global basis. So I think we're at a time right now, so that's kind of history. So what's the future? The future is paying attention to those four things. Joint action is absolutely critical. Having this collaboration between scientists, geneticists, clinicians, patients and families, and advocacy organizations is the only way forward. Having the right tools is absolutely essential, and those tools are rapidly developing in such a way that we're going to reach more people more easily in a more democratic fashion. We have to know the territory. We're understanding HD and the genetic basis much more fully, and that'll probably be fulsome in terms of finding more hidden value. And lastly, and of course not least, is we have to maintain our persistence, because persistence, as they say, always pays. Thanks very much.